Right. So we started the conversation about the infection immunity today. By far, it's one of my favorite chapters in the course. My favorite, of course, is helminths and viruses, but this one's pretty good too. So, few terminological differences. So we're going to talk about microbiome first. This is the first year I pay a lot of attention to microbiome. I should have done it way earlier. So a few terms related to infection and microbiome. When we talk about the microorganisms that cause infection, cause disease, in the overwhelming majority of cases we call them primary pathogens. You may think of, influenza, human immunodeficiency virus, black plague, tularemia, salmonella, shigella as primary pathogens. You understand that, right? So if you get infected with salmonella, you most likely develop salmonellosis. Okay? Those are primary pathogens. Opportunistic pathogens, on the contrary, cause disease only when the host, the infected organism, is immunosuppressed. So we're talking about patients with AIDS, patients in distress, or people that experience immunosuppressive therapy. So in addition to primary and opportunistic pathogens, we also have resident microbiome, composed by resident microorganisms. So these resident microorganisms inhabit a variety of body sites, and we're going to chat about the body sites that do have microbiome. And we're going to mention ones that do not. And this is something that I'm going to ask on the exam. Which of the following body sites has a microbiome or doesn't? Now we acquire these microorganisms, the resident microorganisms, from the very birth, when we pass through the birth canal. Not quite surprisingly, but children that are born through C-section have microbiomes different from children that are born by a natural route. Moreover, there were studies demonstrating that these alterations in the microbiome, because of the birth route, last a lifetime. So even, in adolescence or adulthood, you can still trace, the differences in the microbiome composition to the birth route. We also gain microbiome through exposure to food, environment, people, air, water, all of that kind of stuff. Okay? Does that make sense? Now before we move on to microbiome and questions about it, let's quickly establish the body sites that are always sterile. So this is the list, sterile body sites. We're going to review them. So first, brain. You see the question mark next to it. Generally, we presume that brain does not have any normal resident microorganisms. But several studies demonstrated that in the brains of healthy people, people that didn't die of the brain damage, let's put it this way, okay, you can identify DNA of bacteria in their brains. Of course, the question is it just an artifact, or did this bacteria actually inhabit the brain? Does that make sense so far? Heart. Still a question, because heart by itself is sterile, but blood carries all kinds of microorganisms all the time. So the question is, do we have any of those microorganisms colonizing endothelium of the heart? Do we have the answer? No. So for our intents and purposes, both heart and brain are sterile. Now lungs and lower respiratory tract. By lower respiratory tract we mean trachea and bronchi. Are they sterile? The consensus now, yes they are. There is no, and that's really important term, resident microbiome. When you inhale the air, there are a lot of microorganisms in the air. But those microorganisms get cleared from your lower respiratory tract or lungs by either, being trapped in the mucus and removed with, by ciliary movement, or they're basically destroyed by alveolar macrophages. Make sense? So there's no resident microbiome. Yes, if you take a pulmonary gavage, you can probably find some microorganisms, but they're not gonna stay there. All the glands, liver, gallbladder, spleen, kidneys, urinary bladder, ureters, so basically internal organs, unless it's digestive tract, sterile. Proximal urethra. By proximal urethra, I mean the part of the urethra that is closest to the urinary bladder. The one that opens up into the environment, no. Okay, it is not sterile. Ovaries, testes, muscles, bones, stuff inside your head, inner ear, eye, internal eye, sinuses. Blood. Blood and cerebrospinal fluid, CSF, sterile. No doubt about that. Blood. Again, no resident microbiome. Does that make sense? Now you do have bacteremia, meaning bacteria in the blood, twice a day when you brush your teeth. Or more or less, depends on how often you do it. But every time you brush your teeth, you have bacteremia. Does that make sense? All fluids that are formed in our body, from saliva to urine to sperm to tears, anything that is formed in the exocrine glands, before it leaves the body or leaves the gland, it is sterile. Of course, your saliva in the mouth isn't, but saliva in the salivary glands, absolutely sterile. Okay, urine in the kidneys, sterile. Amniotic fluid. It's a great question. We consider it to be sterile. But again, there are studies showing that you can isolate nucleic acids of at least viruses from amniotic fluid. And on top of that, you know what meconium is. Meconium is basically feces that a newborn baby passes immediately after birth. Okay? So meconium should be sterile because baby doesn't eat. Baby is not exposed to the environment in any way. Well, it turns out you can find quite a few viral species in the meconium. The question, of course, is how did those species get there? There's no answer yet, but probably there's some microbial exposure even in utero. Does that make sense? Good. Now this image shows you main sites with a resident microbiome. Mouth, gut. By gut, it means all the way from the entire alimentary tract. Skin, vagina, supposedly placenta. Okay. Now this image is incomplete. It doesn't take into account, for instance, uterus. Not uterus, sorry. Urethra. Okay, misspoke. So if you would look at this circle, so each circle is divided into several sectors. Each sector corresponds to a particular taxa of microorganisms. For instance, you can see that in the gut, the overwhelming majority of species belong to the class of bacteroides. Okay? In placenta, it is proteobacteria. In vagina, it is lactobacilli. So far, does that make sense? Now, first thing that's kind of captivating our attention in regards to microbiota. Microbiota is different at different body sites. Your oral microbiome is different from your colonic microbiome, and it's different from vaginal microbiome. Got it? Second, it's very stable, meaning that you're going to have two dominant taxa, bacteroides and firmicutes, in the gut. The ratio between those two can change, but not too dramatically, right? Does that make sense? And you're not going to find their enormous amount of lactobacilli out of nowhere. So the general composition of microbiome is fairly stable in a particular body site, and it's stable between people. So if you would survey all of us, you will find practically the same taxa on the skin and in the gut. Okay? I want to apologize. I'm going to take a little break because I noticed that Jeff is there. I'm going to ask for the bottles. All right, so moving on. And does that make sense to you? So microbiome is fairly stable but different at different body sites. Sometimes microorganisms are passing through without establishing residence. We call them transient. Now, what is the example of transient? Have you noticed, if you travel to some significantly different society, I'm not talking that, going from Cleveland to Columbus, but say going from the United States to Kuala Lumpur in Papua New Guinea, or is that Papua New Guinea? Yeah, I think so. Papua New Guinea. Yeah, Jakarta is Indonesia. Or going to South America, okay? And I mean like rural South America. You're going to be exposed to different foods, different water, different level of hygiene. And one of the first things that you may experience is diarrhea or loose stool. That's because of local microorganisms becoming transient, changing the composition of your gut microbiome, changing the transit time, changing all the digestive processes. Does that make sense? That is totally possible. Okay, so these transient microorganisms may be pathogenic, maybe not. Okay, I mentioned before, people sometimes pick up salmonella when they travel. Okay, especially in Central Asia, they pick up salmonella, they get sick, they come back. Salmonella doesn't go away right away. It can stick around for several weeks after people recover. Typical example of transient member of microbiome. Make sense to you? Okay. Now, why do we care about microbiome so much? It is one of the main, I would argue, protections against infectious agents in the human body. If you think about it, resident microorganisms, whether they are on the skin or in the gut or in the mouth, they increase competition for resources, for nutrients, for ecological niches, essentially protecting the host from the primary pathogens. Moreover, as we will see soon, resident microorganisms, members of microbiome, can modulate and shape not only host immune responses, but also post-metabolic profile and diseases that go far beyond the infection. Lastly, if you think about it, you get exposed to a wide variety of microorganisms on a daily basis. You go to the public restroom, you are in the close contact with a bunch of other people, you breathe the same air, you buy food that God knows how it was handled. So all that kind of stuff exposes you to a bunch of microbes. And you're not getting sick that much. Does that make sense? And you will see throughout the course, and you probably have seen already in some of the examples, as long as the microorganism is present in that, microbiome rich environment like gut or mouth, the infection is either mild or limited or non-existent, or you have to be really, really immunocompromised to get it. But once the microorganism enters one of these sterile body sites, the infection sets on right away. Remember we talked about staphylococci. Just a good example. Staphylococci. They're in the skin. I mean, is that a nuisance? Absolutely. Furuncle is the nuisance. Impetigo is the nuisance. But it's a nuisance that isn't a life-threatening nuisance. If this microorganism enters the bloodstream, you're getting endocarditis. And that's not a nuisance. You can die from it. Make sense to you? Okay? Members of human microbiome like Streptococcus agalactiae can enter the baby's bloodstream, end up in the brain, causing meningitis. There you go. Sterile body site, no competition. It's the life-threatening disease. So sterile sites are more prone to the disease than sites with the microbiome. If you would be to pick a site that is more likely to be infected out of the, say, mouth, gut, skin, or brain, despite the fact the brain is, behind the cranium, brain would be the one to pick. You see what I'm trying to say? Because it's sterile. Any sterile body site is more sensitive to the infection. We're good? With this, we are moving to locations of the microbiome. Now, first, I kind of struggled with this decision, whether to make you memorize the members of microbiome or not, decided not to. Because I don't think it facilitates any conceptual learning, just memorization. Like, okay, you now know that the main population in the gut is bacteroides. Well, good for you. What does that mean for you? Probably nothing. Okay? And it's totally normal, unless you study this. So, these are just examples of the most common microorganisms found in the different locations. And you can see that they are very different. Propionibacteria, skin. Okay? Upper respiratory tract, I really like the name. Tenoricutes. It's different mycoplasma. Okay? Oral cavity, Streptococcus is overwhelmingly dominating. Intestine, Bacteroides and Firmicutes. Vagina and urethra, Lactobacilli. Stomach is fairly poorly inhabited. Helicobacter, practically the only species that can survive there. But my point is, if you would look at different diseases associated with the human microbiome, it's actually very telling. Say, take psoriasis. You all know what psoriasis is. Skin disease, right? Turns out that different ratio of firmicutes to actinobacteria in the skin is associated with the psoriasis. I'm going to pick this table apart, not every example we will review. Obesity, the epidemic that currently runs in the United States, reduced ratio of bacteroides to firmicutes. Okay? IBD, inflammatory bowel disease, ulcerative colitis, increased abundance of Enterobacteriaceae. Does that make sense? In colorectal carcinoma, basically the colon cancer, associated with large populations of the species from the genus Fusobacteria. Now, I try to be very careful using the word associated with, because we don't really know if these changes in bacterial populations are causing the disease or if they are simply associated with the disease. Because maybe there is something else, some other underlying mechanism in, say, IBD that facilitates the increased number of Enterobacteriaceae species. You see what I'm trying to say? Now, I wanted to talk about some really interesting aspects of microbiome and human health. And this is just a lovely picture here. Okay. Now, the analysis, first of all, I want to say that analysis of microbiome is extremely complicated, because you have to compare the populations with often hundreds of different taxa, hundreds of different species, and you have to find these changes, in microbial diversity or something else between different body sites or different conditions. So if we're going to talk about skin microbiome first. And I found this wonderful manuscript which shows you the populations of microorganisms at different body locations. And I'd be honest, if I was looking into it, I had no idea that the populations of microbes are so different in different body sites. Look at this. For instance, the head and upper chest, it's one population. You go into the arm, that's the different population, mainly proteobacteria. Okay? You go into the lower abdomen, completely different population. So like umbilicus is entirely populated by Corynebacterium, belly button. Okay? You look at the legs, mainly Staphylococci, which by the way explains why socks smell so terribly. Okay? Because of the fermentation that Staphylococci carry out, producing all the terrible smell. So first of all, even in the terms of a single organ, like a skin, microbial compositions will be different in different locations. And secondly, the study of association between the atopic dermatitis and microbial populations found a strong correlation between the two. So if you would look at the upper graph, it shows you the score of atopic dermatitis on the x-axis and diversity index, the y-axis. So what is score of atopic dermatitis? How bad the dermatitis is. Does that make sense? Shannon diversity index. Shannon diversity index is the parameter that shows you how diverse the population of microorganisms is. How many different taxa you see there. So it turns out that at the baseline without any atopic dermatitis is shown with yellow dots. You have certain distribution of the Shannon diversity index in different subjects. Those are human subjects. During the flare, the diversity of the population changes. It actually decreases. And post-flare, the diversity of the population comes back. So Basically, this graph shows you that during the atopic dermatitis outbreak in a person, the diversity of microorganisms on the skin reduces. Okay? Does that make sense? Now, does that mean that if you increase the diversity, it's going to heal atopic dermatitis? We don't know. It's not a definitive answer. And if you would look at this very colorful image on the bottom, it shows you the relative abundance of different microorganisms on the skin. You see that the majority are Staphylococci, the purple ones, okay? And this is controls. It's probably going to be easier if you look at the controls here. They basically took all other microorganisms out, leaving only Staphylococci to make it more obvious. So this on the left, this image, refers to controls. That's certain abundance. Okay? That's the baseline. Now this is the amount of Staphylococci, this one, in the middle, in the patients without any treatment during the flare. And then you see that post-flare, the bacterial populations come back to normal, and treatment reduces them back to normal. So it kind of shows you that there is a strong association between the number of Staphylococcus species on the skin and atopic dermatitis. Now, does it have any therapeutic potential? It's hard to say. If we can develop some kind of a maybe cream that will, increase the microbial diversity, maybe it will help. If we can look at the microbial population on the skin and diagnose folks that have higher risk for atopic dermatitis, that may help as well. Does that make sense? That's the hard thing about association studies. You cannot take a bunch of people and sterilize their skin. It's not possible. Make sense? I mean, no, it's not possible without killing a person. Now, that's the relative population in the mouth. Just kind of to give you an idea how many taxa we have. Remember we did a mouth swab? We identified streptococci from the class of firmicutes, mainly. They're most abundant. Okay? Turns out that out of the species that were cultivated, this, the percentage that was actually cultivated, less than a third of the firmicutes in the mouth can be grown on the plate. The overwhelming majority of microorganisms that are present in the mouth microbiome are not cultivatable. You cannot grow them on the plate because we don't know the proper conditions. We still can identify them using, say, PCR or deep sequencing methods, but we can't grow them. Does that make sense? What I like the most, unnamed isolates. That's lovely. Meaning that, you grow them, but you don't really know how to call them because they were not discovered before. We didn't give them names. Completely new species. And that manuscript, I don't have a reference here, but that manuscript was published quite recently. It's something like 2011, 2012. So we're talking about discovery of completely new species, less than 10 years ago. Which kind of gives you an idea how much we still don't know about the microbial populations. Now, is there any association between microbes in the mouth and the diseases? Yes. So dental caries is associated with Streptococcus mutans to a large extent. Okay, now, I kind of have a problem with Bifidobacterium being associated with the caries. Again, its association, and Bifidobacterium is the powerful probiotic. It is fermenting, does produce acid, but we need to look into it better. Endodontic diseases. The root canal. Associated with certain species of enterococci from cheese. It was shown that people who eat some types of cheese are more frequently developing endodontic diseases. I don't know if we should, I believe it's Swiss cheese, like that really stinky one. Yeah. But again, the problem is, here's the deal. The problem is, it's the association. Does that make sense? Gingivitis, that's very well known. Fusobacteria, especially Fusobacterium necrophorum, causes gingivitis. The disease can be absolutely devastating. Sometimes it becomes necrotizing, called necrotizing gingivitis. Gums are bleeding terribly, and people lose their teeth, like all of them. Okay. Periodontitis is associated with Porphyromonas gingivalis, and pretty cool story. I don't have it here, but that name, Porphyromonas gingivalis, just kind of brought back some memories. So, you know what periodontitis is? It's the inflammatory disease that destroys the bones, the alveolar processes in which your teeth are sitting. So you may sometimes see people with periodontitis. The root of the tooth is really exposed. The gum line is descending down. Their gums are usually swollen, bleeding. Teeth are not sitting well in the gums, and treatment is either removal of the teeth and installation of dentures, or there's surgical treatment with something with gums. You can do some surgical treatment, which is terribly painful. A friend of ours got this treatment, and when she finally was able to speak, because you cannot talk for like a week or something, and she said if she would know what she would have to go through, she would never agree to this. Like, her teeth were fine, but she said it was terrible. It was awful. Now, why pay so much attention to Porphyromonas gingivalis? One of the recent studies demonstrated the association of Porphyromonas gingivalis with Alzheimer's disease. And not just, it was found in the brain samples of people who died. They compared brains from people with Alzheimer's and people without Alzheimer's, and they found really strong association of Porphyromonas with Alzheimer's. Not only this, they did this experiment in mice. They actually, did the infection of a brain with Porphyromonas, found that Porphyromonas causes Alzheimer's-like syndrome in mice. They managed to identify the mechanism which probably contributes to Alzheimer's in mice. Well, quote-unquote Alzheimer's. Does that make sense? So there's really a good correlation between Porphyromonas abundance in the mouth and the risk for Alzheimer's disease, which gives you an idea. Brush your teeth. Brush your teeth really, really well. Actually, oral health was linked to a variety of conditions, from cardiovascular disease to now Alzheimer's. Okay. Now, gut. That is fantastic. A couple of things about gut microbiome that I just love. So, first and foremost, now that is close to my heart because one of my best friends is doing gut. He used to do gut microbiome, inflammatory bowel disease stuff. And now he studies colon cancer. And my wife works in the lab that studies gut microbiome. So we know that gut microorganisms are necessary for the properly shaped intestinal immune responses, especially intestinal immunity. Now, for instance, germ-free mice, mice that do not have any intestinal microorganisms, have altered and not completely competent immunological responses. Okay, the lymphoid follicles, the Peyer's patches are underdeveloped. The CD8 T cells, the cytotoxic lymphocytes, have lower toxic properties, so they are immunologically inferior to mice with the proper intestinal microbial population. It was actually shown that some bacteria can reduce the symptoms and signs of inflammatory bowel disease. General, like bifidobacteria and lactobacilli, which can be found in products like yogurt, kefir, buttermilk, and other products of milk fermentation. We often call those products probiotic, right? And there are studies showing that probiotics actually reduce the symptoms of intestinal inflammation in people with IBD. Okay, so far, there are several studies showing the role of microorganisms, gut microorganisms, in different clinical syndromes. For instance, I mentioned before stool transplantation as the way to treat Clostridium difficile infection. This is pretty common. Okay. There are studies ongoing on the bacterial intervention to reduce the development of atherosclerosis. Okay. Bacteroides fragilis is used to treat anxiety. Some commensal microorganisms can reduce the risk of developing colon cancer, and genetically engineered E. coli can actually reduce adiposity, meaning that it can reduce the symptoms of obesity. That makes sense so far? On top of that, there are observations that supplementation of probiotics early in life may actually ameliorate the signs of autoimmune disruption of pancreatic islets and either prevent or reduce the insulin deficiency in patients with high risk for type 1 diabetes. Okay. Now, one of the things that kind of, stand close to my heart, I studied obesity for a long time, is the role of intestinal microbiome in obesity. So the graph A shows you the abundance of firmicutes and bacteroidetes. Firmicutes are solid dark columns, dark bars, and bacteroidetes are light bars. And the mice on the regular chow versus Western diet. Now I like the name of the diet. It's called Western diet. Basically, it's a lot of sugar, a lot of cholesterol. Okay. Now you can see that on Western diet. By the way, it tastes really good. I tried it. It's like cookie dough. Okay. So on Western diet, mice accumulate much more significant amount of fat. Okay, they become obese. And you can see that they not only become obese, the proportion of firmicutes to bacteroidetes increases. So they get more firmicutes, less bacteroidetes. Okay, does that make sense? They also get more molicutes. You can see that in Western diet mice increase the percentage of body fat. Does that make sense? And you can see that mice on the Western diet have lower microbial diversity in the gut. So this Shannon index represents the diversity of microorganisms. You see that in general, mice on the Western diet have a lower level of microbial diversity compared to the mice on the regular diet. So what does it tell you? First, things that we really want to jump to conclusions here. So conclusion number one that we want to jump to: increased firmicutes promote obesity. Right? Does that make sense? Is that so? It's hard to say. I'll get back to this notion later. Second, increased diversity of the population, of microbial population, is good. Again, is increased diversity does it reduce the risk of obesity, or just an association? Now, how can we test if actually microbes facilitate the development of obesity? We can take mice that are germ-free, feed them with Western diet, and then transplant the microbiome from lean mice and see what happens. Does that make sense? So you take mice that do not have any microbes, but that are obese, and you give them microbes from regular lean mice. You got it? They lose weight. Now, if you do the opposite, and you take microbes from obese mice and put them in the lean, germ-free animals, they will start gain weight even on the regular diet. So they will experience different feeding patterns and different utilization of energy. So there is not only simple association, there is also some kind of a, microbiome facilitating the, weight gain. We're clear. Now, that is a pretty cool part. Another part that kind of forces us to think, it's not in the image, but I'm kind of giving you an idea. You've all heard, for a long time, FDA and CDC, they were promoting a low-fat, high-carbohydrate diet, right? Fat is bad. It's very likely that this is what contributed to the epidemic of obesity in the United States. Because it turns out that if you would compare mice or people, these experiments were done in people. Let's say people who eat high carbohydrate, low fat diet will have higher percentage of firmicutes and lower percentage of bacteroides. Does that make sense? That's high carb. People who are on the high fat diet will have this completely the opposite. Now, if we come back to this graph, you can see that higher percentage of firmicutes is associated with a higher percentage of body fat. The obesity. Does that make sense? Now, the implications are very complex. It's really far out there. We cannot say, oh, just give them a ton of bacteroides and everything's going to be fine, because we don't know what's going on there. Maybe it's the energy utilization that firmicutes use. Couple more things I wanted to say about this two populations, firmicutes versus bacteroides. If you take a mouse or a person, okay, say you have a person that consistently eats high carbohydrate, low fat diet. Make sense? Normal weight, just high carbs. And you put this person abruptly on the high fat, low carb. You flip the diet. Does that make sense? You understand what I'm saying, right? you will notice the change in the bacterial population. High bacteroidetes, low firmicutes in 24 to 48 hours. So the change happens almost immediately. Your gut microbiome responds very quickly. Okay? Not only this, there was a study that was looking at the diversity of gut microbiome in the normal weight people. And it turns out that increased diversity, which from ecological standpoint is always good. More diverse populations are more stable. Does that make sense? Nutritionally stable, just stable. Increased diversity is strongly associated with the amount of fiber that the person consumes. Again, if you read popular press, you may find, oh, scientists uncovered, the magic trick how to treat, I don't know, obesity or diabetes. Diabetes is the same story, basically. Diabetes in people using microbiome. It's not that simple. We have a lot of association studies, okay? And this association study is basically saying okay, if you eat a lot of fiber, fruits and vegetables, a lot of fat and protein, and very little carbohydrate, you're going to maintain healthy microbiome and stay lean. Duh, I didn't need the entire microbiome being sequenced to, find out this answer. Does that make sense? So it's kind of, goes in line with the most recent nutritional recommendation, blah, blah, blah. But it still would be interesting if we can do some kind of intervention to maybe reduce, say, the calorie utilization by the intestinal microbiome. Does that make sense? I learned about it recently. Okay? Now this is really awesome. First set of graphs that I wanted to show is on the bottom. Now I'm going to break it down very simple. It's on the left, two old friends, firmicutes and bacteroidetes, in the guts of the patients with autism spectrum disorder and neurotypical patients. So you see that in patients with autism spectrum disorder, the firmicute population is much higher than in patients who are neurotypical. Neurotypical is the term that, describes people without autism spectrum disorder. Does that make sense? This graph on the right shows you the only significantly different population, bacteroidetes, in neurotypical patients are significantly higher than in autistic patients. So, cool, I suppose. Are you going to feed them bacteroidetes? Which is not feasible, to be honest, because bacteroidetes are obligate anaerobes. I have no idea how you can do that. So, again, cool. This year, I think it's this year. I mean, we can go and check the link. I have no clue. Yeah, well, whatever. I think it's 2019 it was published, but basically the study was done last year. There's a group, I think in Italy, that do really interesting studies on the role of gut microbiome and development of autistic behavior in mice. Because that's, the model. So what they've done, they have collected several different models for autism in mice. I don't know how to highlight it more for you as future health, professionals. Mouse models. Mice are not men. Okay? mice are not human, they are models. We have models of autism disorder in mice. They're not conversationalists. You cannot figure out whether they're autistic or not by talking to mice. So it's called, social behavior deficit. Makes sense, right? They're different models. So what they've done, they've collected these models, different models, and they fed mice with one species of bacteria, Lactobacillus reuteri. So that's your example, so your mouse models, really cute mice, they drink Lactobacillus reuteri and they become less autistic. Okay? No, seriously, it reduced the asocial behavior, made mice more social in different independent models. Not only they showed, and that would be cool by itself, but they also showed that first, it's mediated by the vagus nerve. They basically cut the vagus nerve in mice and see what happens. That's it. If you cut the vagus nerve, there's no response. So obviously something happens in the gut that sends the signal via vagus nerve all the way to the brain, brain stem, and then from brain stem something, happens. And then, I don't know, maybe there's some kind of a response back. Moreover, they were curious which chemical is responsible, which neurotransmitter. And it turns out, it's oxytocin. They knocked out oxytocin receptor. And when they knock out oxytocin receptor, Lactobacillus reuteri loses its efficiency. Now, can you buy Lactobacillus reuteri? Online, about $8 a bottle, the pills. FDA approved. I mean, it's not a drug, but FDA tested it and said you're not going to die from it. And usually it's present in milk fermentation products. You can read the list of bacteria and yogurts and stuff like that, So, the authors of that paper actually very careful in claiming that, we found the treatment for autism. They say that, we should proceed to human studies, but we should be very careful because we do not know what interactions are going to happen in the human gut compared to the mouse gut. Okay, so that's really important. And it was shown to work in the germ-free mice. But again, humans are not germ-free, so they can be. I expect clinical trials coming up really, really soon. Yes?
So do you think they'll ever do something where they could use that for prevention of autism if pregnant women take it?
That's a fantastic question. I don't think there were, as far as I know, there were no studies in treating autism by giving it to pregnant mothers, even pregnant mouse mothers. Possible, I would say, but what they do here, they already developed one. I would say they might actually give it to children at the higher risk of autism, because you can test a child who is like 3-4 months old for composition of intestinal microbiome if there would be developed if clinical cutoffs will be developed, saying this kid has certain percentage of firmicutes, this kid is at risk for developing autism, let's give him lactobacilli. That I can see in fairly nearest future. Any experiments on pregnant women? Nobody wants to touch it with a 6-foot pole. Okay? Even kids are easier. Because, if the kid is sick, parents will consent. When woman is pregnant, it's kind of a high-risk area because, if it affects pregnancy, that's a huge problem. Might be. Hold on. My point is, you're going to find a bunch of lactobacilli in things like buttermilk. Or, kefir, which is kind of a fancy, expensive version of buttermilk. They taste good. Like, drink them. They're not going to hurt you, just, drink them. They're good.
I was just wondering, when are they thinking that autism develops? Is it something that develops in the womb or as the child develops? Because you're asking because you're thinking of preventing it, but do they even know when?
I don't. That's the question out of my competency.
That's why I wonder.
I understand your question, and it's a great question, whether the child is born autistic or kid is developing autism with time.
They don't like to diagnose it before 2 years old, but you can see signs as early as 6 months.
So
There's also been people who are 8 and aren't diagnosed with it until they're like 8 or 9.
Yeah, it's just
Yeah.
depends on the person. Everybody is on a different. Every child that's been diagnosed is on a different spectrum and usually
Yes.
at a different time that they've been diagnosed.
now, my comment, and I was lucky not to deal with autism in my own family, okay? But we now don't call it autism, we call it autism spectrum disorder, because it can be different degree of asocial behavior. Does that make sense? So I remember some kids from my childhood and my school experience, and I bet some of them now would be diagnosed. And they were either, back then they were scolded by teachers for not doing well in class, or bullied by peers for being weird, and they were considered to be just, weird kids. So when, people say, oh, autism is on the rise, no, our ability to diagnose it is on the rise, okay? Like every year, CDC, well, not every year, but from time to time, CDC will reconsider the diagnostic criteria for obesity, and then Louisiana loses its honorable first place in being the most obese state in the U.S., and some weird West Virginia comes forward. So a lot of it is diagnostic approach. But those two questions, answer my take on your question, just drink buttermilk or eat yogurt. It's just good. It's not going to hurt. Exactly. And to your question, I don't know. Sorry.
Okay.
I will look into it and will try to give you an answer on Monday or some type of answer. Now, this unit consists of two separate parts, infection and immunity. Infection is, I like both, but I kind of, fair warning, infection is a lot of terminology. A lot of just terminology. Knowing what the words mean. Immunity, there are terms as well, but there are a lot of conceptual understanding. So, first, what is infection? Infection means replication. Always. If there's no replication, there's no infection. Infection, meaning replication of pathogen, does not necessarily lead to the disease. In this case, we call it asymptomatic. How many times you had a situation when you are the only person in the family that did not get the flu? It can happen. Does that mean you weren't infected? No. You may have been infected, you just never developed symptoms. Virus replicated, but your immune system crushed it early. Infectious disease, and that's important. Remember, we're talking about infectious disease. Disease that is caused by a microorganism. Infectious disease can be communicable. Obviously. Flu, common cold, HIV, meningitis. Does that make sense? Sometimes it is non-communicable. Take peritonitis, the complication of appendicitis, when appendix bursts and all those microorganisms end up in your peritoneal cavity. Am I clear? There you go. It's not communicable. You cannot infect another person with your gut microbes. Okay? Or primary amoebic meningoencephalitis, or granulomatous, doesn't matter. Amoebic invasion, we talked about Naegleria fowleri, brain-eating amoeba. You cannot infect another person with it if you have it. So it's a non-communicable disease. Does that make sense? Another word that we use for infectious or communicable is contagious, which sounds really sort of a layman term, but often scientists use the term highly contagious disease, which means highly transmissible or low contagiousness. Does that make sense? Now, oh, yeah. How can you characterize the contagiousness? There is a parameter called the basic reproduction number. The table, the left bottom corner, shows you the basic reproduction numbers, or R0s, for different infections. Now, few words about basic reproduction number. First, what is the main idea behind it? Basic reproduction number, technically, shows how many people will be infected by a single sick person. You understand the idea? So for instance, if somebody has flu, this person normally will infect 2 to 3 other people during the course of disease. Does that make sense? Now, a couple more comments on what basic reproduction number represents. First, it's a model. It is not measured. You got it? It's not measured. It's a model. If I have flu, I found out, say, I have influenza and I really sit at home all the time, and I got it from one visit to Walmart, and then I sit home for 3 weeks, I'm not going to transmit it to anyone during these 3 weeks. Does that make sense? Now, if I have flu and I walk around and I cough and I speed and I sneeze on every doorknob and faucet possible in Lakeland, I will transmit it to a hell lot of people. Does that make sense? We're talking about model, how contagious the disease is. Second, basic reproduction number has nothing to do with the severity of infection. Look at this. Ebola is a basic reproduction number around 2. And it's really terrible disease, right? You can die from it. On the other hand, I don't know, something mild. Well, let's say mumps. This basic reproduction number in the range 4 to 7. And, I'd rather have mumps than Ebola. Okay? The most contagious disease that we know of is measles. And I'm not even going to start on vaccination. Just let me give you an idea. I can compare measles and Ebola in terms of the contagiousness. Let's say you all are not vaccinated against Ebola, and I have Ebola. We can do an entire class with the condition that I'm not coming close, I'm not spitting on you, vomiting, peeing, or touching you during the class, and you'll find it. And nobody's going to get infected. Does that make sense? Second modal situation, you all are not vaccinated against measles, and I have measles. I walk in the class, we spend 3 hours, everybody's infected by the time we leave. Actually, everybody's infected in about an hour. Measles is extremely contagious, unbelievably contagious. You can pick it up from walking by a person who has measles. Okay, just walking by on the street. Make sense? So, this is what basic reproduction number describes. It's a model that shows how contagious the disease is. Yes?
Since the MMR vaccine is really the only one that comes live virus,
Uh-huh.
how do you not become contagious when you get the vaccine?
Because virus does not replicate to the levels high enough to infect other people.
Okay.
Does that make sense?
I got it yesterday.
Actually,
so I really wanted
good.
to know.
Actually, polio vaccine, one of the ideas behind polio vaccination, live polio vaccine, the one that is administered, it's not common in the United States. You get weak virus, you pass it with feces, it enters the environment, and this weak virus, non-pathogenic virus, can then be transmitted to other people, basically vaccinating them without any medical contribution. Okay, it's done mostly in the countries where logistics is an issue. Does that make sense? Nosocomial infection, hospital-acquired infection. These infections are often opportunistic, like Serratia marcescens. Yes.
Which disease did you vaccinate off the plan? Was that smallpox?
Yeah, smallpox is gone.
I just couldn't remember because I know polio's up there, but
Polio is, we're going to be there eventually. Smallpox is gone. We don't have smallpox on the face of Earth. Okay, it's totally gone. And we can eliminate rubella, mumps, and measles if we would be that little smarter, because all those infections are entirely human. So all we need to do is to vaccinate every single person on Earth. Every person. Okay? And then it's gone.
Got too many idiots, that's not
Yeah, unfortunately. Now, this is the table showing you. So what I hate about current data presentation is people got so many tools about data presentation that it's really hard to find a good old table, like table or graph, okay, to demonstrate. So basically that's the standardized infection ratio, okay? So this first column shows the change in 2016 versus 2015 in hospital-acquired infections. And I can tell you we're doing all right. Okay, you see that all hospital-acquired infections are on the decline. We're doing better in preventing those. So, urinary tract infections, bloodstream infections, surgical site infections, MRSA, respiratory infection, ventilator associated events, they all are on the decline, which is good. Not only this, we are lowering the rate of infections compared to national baseline, which again is good. Okay, so hospital-acquired infections are going down, but they still are a very real threat. Now, iatrogenic infection is the result of medical procedure. And this I want to make a very clear distinction between nosocomial and iatrogenic. If you go to the hospital for, shoulder replacement. You go for shoulder replacement and you end up with UTI, it has nothing to do with a shoulder replacement. This infection happened because you were in the hospital. Does that make sense? That's nosocomial. You see? If you get bone marrow transplant, and it turns out that bone marrow of a donor had a latent Epstein-Barr infection, and you develop life-threatening Epstein-Barr infection after bone marrow transplant, that's iatrogenic, because that's the direct result of a procedure. Does that make sense? Shoulder replacement, infection of a surgical site because the prosthetic joint was dirty, that's iatrogenic. That's the result of a procedure, direct result of a procedure. Obviously, iatrogenic infections are less frequent. Nosocomial just more common because people just spend time in the hospitals.
they're common with implants.
Of course. I'm talking about like incidence overall. Finally, zoonotic infection acquired from animals. We have a bunch of zoonotic infections, from West Nile virus or yellow fever to some types of influenza. Okay, so we're going to wrap it up.